first of all, I would like to use the opportunity to say thank you to Fred. I don't see him here, but I know that he has his people here. Because without Fred Kavli, we wouldn't be able to have this exciting event this week, to celebrate science. And I think that is so important. Of course, he has a lot of support as a government and the academy, but it's this man's idea and money and his driving force letting us celebrate. And I think that is fantastic. Okay, so the title of my talk today is A Taste of Memory. And that is, of course, because there will be a lot of food afterwards, but I'm not going to talk that much about food. But I think the main message here with the taste of memory is that memory is such a big field, so I could spend a week with you telling about all we know about memory, but since I have only 45 minutes, I can give you a taste with the highlights from this field. So I want to start by just playing a very cute video for you. Yeah. Enjoy the rat chef. Here is the restaurant critics. This is the boss.
They must be joking.
Look at him. This is not good. He's sent back in time.
Who cooks the ratatouille? I demand to know!
Okay, it goes on, so we can stop it here. It's okay, we can take that. That's so nice.
To give my compliments to the
Who
chef.
is the chef?
And now I find
It's a
myself
rat.
in the extraordinary position of having my waiter be the chef.
Thank you. I'm just your waiter tonight.
Then who do I thank for the meal?
Excuse me a minute?
Okay, so now he's, of course, very embarrassed because he has to tell my chef is a rat. But what I wanted to illustrate here is that taste gives you memories. You can run in time back to when you experienced this taste. And one of my friends, John Aggleton, visited a wonderful museum in York that I haven't been visiting. It's a Viking Museum. And when he came back from that museum, he decided, I'm going to test this in the lab. So what did he experience in this Viking Museum? How many of you have been at York Viking Museum? Oh, so you know what I'm talking about. I don't know, but, but there are, so what they say is that they have the Vikings there, they have a lot of sounds and violence and houses and so on, but they also have odors. So I think the Vikings were not smelling French perfume. So what John Aggleton did, and I got the slides from him, is to bring these odors to the lab. And then he asked, if you get this odor, can you also remember other things from this experience? And we can see what happened. So this group of people, they got a lot of questions about this museum. So mean correct here is that they're doing quite well. So this group, they got odors from this Viking Museum here, and afterwards they just got a random odor, and still they did quite well. What about those people who hadn't been to this museum for maybe 10 years? And they got the control order here, then they didn't remember anything. Then they got the Viking orders, and suddenly they got back the memory. Just like we saw in the restaurant from the tiny movie that I showed you. So odors and taste are quite important for our memory. So is it so that these odors and taste, do they trigger a blueprint from our memory? Is it like we are playing a video from our lives, from our experiences? Or how is memory organized? I'm going to show you a tiny clip from a television series that is called 60 Minutes that Americans probably know. I don't know that much about it, but this one I think is quite amazing. So this video that I'm going to show illustrates that memory is not a blueprint. So it is about a woman who is raped. And she's thinking for herself, if I ever survive this rape, I'm going to remember this man. And he is going to be in prison as long as I live. So her motivation for coding in his face is enormous. So here we can see the video. I'm going to stop it in the middle, but just get the flavor of it.
It's a cliche of courtroom dramas. That moment when the eyewitness is asked, "Do you see the person who committed the crime here in this courtroom before you?" Well, it happens in real courtrooms all the time. And to jurors, that point of the finger by a confident, damning as evidence can get. But there is one type of evidence that's even more persuasive, and that, of course, is DNA. There have been 233 people exonerated by DNA in this country, and now a stunning has emerged. More than 3/4 of them were sent to prison, at least in part, because an eyewitness pointed a finger. An eyewitness was wrong. She vowed to stay alert and study him so that if she lived, she could help.
What is his voice? Does he have an accent? Does he have a scar? Is there a tattoo?
He's raping you and you're studying his face.
It was just trying to pay attention to a detail that if I survived, and that was my plan, I'd be able to help the police catch him.
He was locked up and days later put in a...
You scared?
I was very scared, nervous. I was so nervous. I was trembling. You know, I felt my body just shaking.
They were asked to step forward, speak, and step back.
I can remember looking to the detective and saying, it's between 4 and 5, can I have them do it again?
Okay, I stop it here, but I can tell the rest what happened. So you saw this man. He was sitting in prison at least for 11 years. He was innocent. And she was coding this memory so well, she thought. But she was misguided because the detective thought, this is the man, and lined him up, and then she thought, oh, he looks quite familiar. What happened was also that he arranged that she met the rapist. But her memory of this man that she had created herself was so strong that even when she saw the rapist and the one that was in prison already, she said, no, it's the innocent man. And it was terrible. So what happened in the end is that DNA showed he was innocent. So how does our memory work so that she can be so sure? So now, of course, they are traveling around and telling this story. Please, eyewitnesses are good, but they can be fooled, even though they're highly motivated. So we need to study memory, how memory is constructed in order to not do such failures, to trust eyewitnesses blindly. So how can we study memory? So Lisa Randall said that it's so difficult to study because you need the tools, you need the models, you need ideas. And that's the same in neuroscience. If you don't have ideas, if you don't have tools, you can't do anything. So we have a brain, and we want to find out how is this brain making memories. We can play a tiny video that I got from NRK, seeing all these billions, 100 billion cells talking to each other. And they're talking to an average of 10,000 other friends. So how can we understand these processes in the brain? It's a big challenge. But we have to start somewhere. And where can we start? We can start with patients. And that happened in 1953. There was a patient, HM, or Henrik Gustav Molaisen. We're allowed to know his name because he died a few years ago. He had epileptic seizures, and he couldn't work. He had really big problems because the drugs didn't help him. So the only help he could get is from the surgeon. And the surgeon had to do experimental surgery. Can you imagine doing experimental surgery? He didn't know exactly what would happen, but still, that was Henry's only hope, to have a normal life. So what Scoville did was to remove parts of the medial entorhinal cortex, just inside our ears and on the underside here. So this is under the brain. And he removed a tiny structure that is called the hippocampus. So when you dissect out the hippocampus from a human brain, it looks like a hippocampus, a seahorse, and that's why it has this name. And he also removed a part of the cortex that surrounded the hippocampus. What happened after the surgery is that everything went well, no infections, nothing. HM, his intellectual capabilities increased because his IQ was not disturbed by seizures. He was social, but when the doctors talked to him, he no longer recognized the hospital staff. He couldn't find his way to the bathroom. He didn't remember anything what happened in the hospital. It was like everything happened for the first time. So then Scoville called his friend Penfield and said, "Do you have somebody who can help me to study this person? Because I don't know what happened. I just removed these two tiny pieces in HM's brain. And, sorry, and this is the result." So then Penfield had a student, Brenda Milner, and she said, "Okay, I know what to do. I'm a psychologist, so I have some tests." So she was testing him and found out that he had problems with, for example, remember a story. So if I tell you a fairy tale, a Norwegian fairy tale of the troll and the boy who cheated the troll so that the troll exploded by eating so much food, and the boy just put the food into his pockets, then you will remember. But HM could not remember because he had lost these tiny pieces of his brain. And the same with drawing. If he was drawing and copying a complicated drawing, just 5 minutes after, he couldn't remember anything from this drawing. What is interesting is that these results are from other patients who had other kinds of accidents, also losing this tiny structure, the hippocampus on both sides. And they have big, big problems like HM, and the only structure that was damaged in, for example, RB was this hippocampus. So you see the scores here are quite low compared to control people. I almost said animals, sorry, I'm used to working with rats. So, but what was really interesting is that we think of memory as daily episodes that we remember and that we can remember stories and so on. But memory is so much more. And what happened is that Brenda Milner tested HM on different types of memories. For example, she was teaching him how he could draw a star and follow the star here while just looking into a mirror. I don't know if you tried it, but it's not easy. And if you see the curves, this is on day 1. He has a lot of errors, but then it's reduced during the day. And then he's not so good the next day, but on day 3, he's perfect. So he can learn these things. But then Brenda Milner asked him, do you know how to draw in mirror? No, I've never done it before. Because he can't remember episodes. But still his brain is capable to learn other skills. So now we know that the hippocampus, and I want you to remember that word, hippocampus. So when I see you outside, I want you to say hippocampus. So now we know that hippocampus is very central for episodic memory. So then we can go into the structure and ask, what are the cellular mechanisms for making memories? But before we go into the mammalian brain, we have to visit a smaller brain, the aplysia. And I don't know if some of you recognize Eric Kandel, one of the Nobel laureates. He got a Nobel Prize for his studies on this slug, sea slug, the aplysia. So what Eric started on with his colleagues in the 60s was to study this sea slug. This sea slug has very simple behavior. It's just when you touch him, he's just doing like this because he's, you can say, scared. He wants to avoid being touched. And then, since aplysia has so big cells, Eric and his colleagues could put tiny electrodes into the cells of this sea slug when the sea slug was learning to avoid this stimulus. And this, the sensory neuron got the information of the outside of the body, or the touch of the body, and then the motor neuron, with some neurons in between, of course, sent signals to the muscles in the sea slug. You have to change your body posture, because this might be dangerous. What was important with these studies is that the conclusion was that the learning depends on what happens in the communication between these cells. So if the aplysia is thinking, oh, it's not dangerous, just water or something, then he's relaxing, and then the activity here, or the synapse strength is reduced. But if it's dangerous, then it's strengthened. And that we can see on the next slide. So this is the response from the sensoric neuron, just getting the touch, recording the touch. And this is the motor neuron. And here you see that the motor neuron is increasing a tiny bit compared to what it was before learning. So this is sensitivation. And here we can, they have quantified it. So in the control situation, it's a low retraction, and here it's more after learning. And over days, they could even use the microscope to see the changes on these actual cells. So if the sea slug learned to retract his body very, very hard, then he got more contacts between the cells. And if he just habituated or ignored the touch, then he got fewer contacts between the sensoric neuron and the motor neuron. So then these experiments were extremely important for understanding these cellular mechanisms, and they could start to study the biochemistry and all these things. But our goal is to understand the human brain, so we can't deal with only aplysia. We have to ask the question, can we see similar things in the mammalian brain? And just the next house to here, there was a lab by Per Andersen, my supervisor, is Per, and his student, Terje Lømo, at that time, and Tim Bliss. Terje Lømo started his PhD in Per Andersen's lab, and he was, together with Per, trying to understand what happens with cells when you play with them, when you just stimulate them. And you know that in the brain, the cells talk to each other by sending small electrical pulses from one cell to the next cell. This you can do with just taking electrical current from the socket, just smaller. And that is what they did. So they send in one pulse here into the hippocampus, and then they record the result. And then Per said to Terje, I want you to play. You should have more pulses in a second than only one. And then afterwards, you should again measure the result. So then we can see what happened in that experiment. So this is the tiny pulse. So here Terje is stimulating and then recording. Then he gives this high-frequency stimulation, 20 to 100 pulses in a second. And it's a big response to the same tiny electrical pulse. And Tim Bliss came and joined the group, and they could publish a beautiful paper in Journal of Physiology in 1973, where they could demonstrate exactly this phenomena, that there is long-term potentiation in these synapses. Like we saw in the aplysia, the synapses, the cells are talking more efficiently to each other. So this is the control site, and you just get these tiny bumps, bump, 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 bump. But here, at the stimulated site, it's just increasing. But what is also important, one thing is here that it's specific to those cells that are stimulated, those fibers that are stimulated, but even at this time, they saw that this happened at a time scale long enough to be potentially useful for information storage. And information storage is memory. So this is the mechanism for storing memory. And I can say that because I don't have time to tell you all the research that happened after this, but that has been enormous amount of research supporting that this is the central mechanism for storing memory. So what do we use the LTP for in hippocampus? So this is a beautiful drawing of Ramon y Cajal. And if you just think about these nerve cells that he was drawing here as circles, so this is cell 1, cell 2, cell 3, cell 4, and so on, then LTP can connect, making more efficient communication between some neurons that are stimulated with this high-frequency stimulation, and those cells that are not stimulated do not communicate that well. Do you get that? So if you think about the memory in the hippocampus, we can pretend that it looks like this. Do you agree with that? So, if the memory looks like this, so the memory is encoded on these cells. So the electrical communication between these cells, that's the memory. So this is what we learned when we saw the movie today. But if I tell you, for example, the fairy tale again with the boy, and I don't mention the troll, you suddenly remember the troll and the food and everything. Why do you do that? Because that information is stored in your brain between the cells. But I told you only the boy, so I told you I activated only these 3 small cells in your memory. So because these cells are connected with long-term potentiation, found here in Norway, then the whole memory is activated. And this is a process that David Marr, doing theory, like Lisa Randall, not the same topic though, suggested happens in the brain. It's called pattern completion. So you just fill out a pattern. And I can demonstrate this, that memory or retrieval of memory is a construction process. So I give you 1. Do you see what it is? 2. You don't see? Sorry? It's a cat? Almost. It's a rat. So you see, you would not be then a good eyewitness if you had to say it's the rat, not the cat, because you had too few cues. So that is the danger, of course, with memory. If you sort of don't have enough information, then you think it's a cat and not a rat. But you were good. So I'm doing animal research, and can my research help us to understand memory? What can I do with my rats? So we can try to find out how the cells in the hippocampus in a rat is responding to the environment. This was done by one of my supervisors, John O'Keefe, in London, and he discovered a fantastic cell, a place cell, that I'm going to demonstrate here in a video. But what is important with the place cells is that they are active only when the rat or the human is at a certain place, but it's silent when the person or the organism is at a different place. See if we can get this movie running. So here is Laura Colgin. She worked in my lab earlier. Oh. And this is the rat. The rat is motivated by eating chocolate. And rats love chocolate. So Laura is now throwing chocolate around here. And the rat is just pretend to be a hoover. So he is eating chocolate. And what you will see is that the single cell that we listen to is active only when the rat was in that corner. Did you see that? So whenever the rat was here, it wasn't active. And that we can see here. So this is the trace of the animal, the black, and red is the activity of one single cell in the hippocampus. Here's another cell in the hippocampus, and this one is active when the rat is here at west. This cell is active when the rat is here at north. And we can also make these rate plots, just showing that blue means zero activity and warm colors is high frequent activity. So Bruce McNaughton and Carol Barnes the lab of Bruce McNaughton and Carol Barnes, Matt Wilson worked with them, and they published a beautiful study here where they recorded 80 neurons in the hippocampus. So if you then can think about, this is a box, and the rat is running around here like it did in the video, and this is an example of one silent cell, another silent cell, but here is some activity, and here is some activity. So you see that they were able to record place cell activity of 80 cells at the same time. And what is important here is that they could use this activity to predict the position of the rat in the environment with 1 centimeter precision, just by using the information from these places. So John O'Keefe and Nadel wrote a book and said, this is a cognitive map, what these cells do. Okay, so David Marr suggested pattern completion. Can we see evidence of pattern completion in these cells? Because these place cells are recorded in the hippocampus, and that is where we need to see pattern completion if they are then dealing with memory. So this experiment was done by Jim Knierim and his students. And it looks a bit difficult, but I will try to explain it in an easy way. So the rat is running here on a circular track, just eating chocolate. And around the circular track, there are a lot of landmarks. And in one situation, they rotate the curtains around with some landmarks and then leave some other landmarks stable. And even though there is a change then in the sensoric input to these rats, they keep the same map. And that is what is shown here. So this is the circular track, 0 to 360 degrees, correlated with itself. And for you, you don't need to understand this, that it's a population vectors and so on. But if you could be so kind to just look at the colors here, and if you agree with me that it looks very similar to this map, you agree with me that this means pattern completion. Even though they rotated some cues in this maze, you see exactly the same expression in these CA3's place cells. But then we have a problem. If these cells don't react to big changes in the environment and only do pattern completion, don't we just mix up all memories, rats and cats and whatever we should remember? It's just a mess. So how can the brain deal with that mess? Because if we have only pattern completion, we are lost. Then we can't remember, we can't distinguish episodes. And episodes that we would like to distinguish are different birthday parties, for example. So again, David Marr suggested you can get pattern separation, not pattern completion, but pattern separation by having different cells active. So you remember, this could be one memory. The green cells are active. This is a second memory for this birthday that happened in 2007, and there are different cells active at this time. So this is a good theory. So then we can ask, is there any evidence for pattern separation in the hippocampus when we record these cells? So then we would expect to have many, many maps over the environment when we record these cells. And that is, in fact, what we find. So if we expose the rats to different rooms in the lab, they have to make a spatial map for each room. And what we can show is that cells that are active in one room are not active in the other room, or if they are active in a different place in that room. So by storing the memory on different cells, we have pattern separation, but since the cells within one memory are connected, we have pattern completion. So then we have both. And this, I think this is the most complicated figure, but it's just an idea. So the way we can get both pattern separation, pattern completion, the way we can remember just by having a small amount of cues to get pattern completion and then to not mix them up is suggested by attractor networks. So if you think about the memory as a ball rolling in a crater, that is the memory that, for example, for the boy and the troll. But then we have to differentiate that memory from Ratatouille, the movie. And there are several craters with different memories. And in order to get the ball to move from one crater to the next crater, there has to be a lot of energy, and then you get pattern separation. Okay, I see that this is a bit difficult. We can have something that is nicer. Is this okay? What do you see? You see, oh, you can say a cat now. You see a rat as well. So, this is the essence of the attractor networks. You have a cat or you have a rat, but you can't see them at the same time. Exactly like the rat is in one crater and the cat is in another crater. Do you see that? This we can study in the lab. We can bring the cat and the rat to the lab. We can train the rats of two different shapes of these boxes. So this is a 1 by 1 meter box, and then there is a smaller cylinder. The rat is just walking around, eating chocolate in both of them, but is able to distinguish them. So we can call this is the cat box, and this is the rat box. What happens if you start to morph these two boxes so that it's not certain whether it's a square or a circle? Do you see a cat then, or a rat? Do you see a square, or do you see a circle? And with these attractor networks that I just told you about, in fact, what happens is that all cells have maps here for the square, for the almost square, for the almost square that are similar, but then there's a jump. This cell decides this is a circle. This cell changes its activity here, decides it's a circle. Pull up the circle map. This cell does the same. This cell does the same, and so on. So you see, it is these two separated maps, even though we have a lot of pattern completion in between. So how is memory organized in time? We did some science fiction in the lab using what I said now, that there are footprints or signatures of the different spatial maps, and then we can play in the lab. What we want to see here is how the hippocampus is switching between these different maps, or memories, if you would like. It's called a teleportation task. So the task is so that the rat is developing two different maps. One for this box, which has light at the floor, and one that has light here at the wall, and they have 2 separate maps. So then we can ask, what happens if the rat is in this environment? And since these cues are just light, we can play and we can just put the switch on so that when the rat is in this environment, we can teleport the rat to the other environment without the rat knowing. And that is exactly what we did in the teleportation. So since I don't have that much time, I can tell you the result. What happened is that these maps are separated in time at the theta cycle that you don't understand that much of, but you can measure EEG. So when people have epileptic seizures, you can measure EEG and also in control situations. And that is just that the cells are active, and then they stop to be active, then they're active, and they stop to be active. And it seems like the hippocampus is separating these full memories in these tiny, tiny chunks, quantal memories of 125 milliseconds each. Only one memory at a time. And even at this tiny time scale, they don't mix up memories. So I have 5 minutes left, so we can see how the memories depend on space. We've just learned now about the place cell that John O'Keefe and his student discovered. We ask now, how does the information going into the hippocampus look like? And in 2005, we discovered a cell that is called a grid cell, and that was done with our students, Torkel Hafting and Marianne Fyn and Sturla Molden. So what was so fascinating with this cell is that for people doing mathematics, you see that you can fit in an equilateral triangle on this pattern. And to make a story very, very short, because I don't have that much time, what we think by just doing models is that the hippocampal cell is a linear summation of the grid cells. So if you then think of the different phases here, and you just put the grid cell of different phase length here on top of each other, then you get one single field, and that is what you see in the hippocampus. What we can then ask is that the pattern separation that we see on memories in the hippocampus, is that possible that it happens because something happens on the input side of these grid cells? So here are some experiments that are not published yet from our lab, Hanne Stensola and Tor Stensola and Trygve Solstad and Christian Frøland. So what it looks like is that the grid map is changing in the brain of the rat. So the resolution that we heard earlier today is very high at the dorsal part of the entorhinal cortex. So this is a rat brain, this is the hippocampus, this is the entorhinal cortex, and here are the electrodes recording these cells. And you see that the resolution here at the dorsal part of the entorhinal cortex is quite high, and then you go deep into structure, and then it's like blowing up a balloon where you have put these grid dots on the top, and then you just blow up and it gets bigger and bigger. But what is important is that if you blow up a balloon, then it's smooth. What happens in this situation is that it's not smooth, it jumps. And what is exciting about that is what you see here, grid size at the y-axis, and then you go deeper and deeper, and then it's jumping. the size. And then you go deeper, and then it's jumping again. So then we can ask whether these grid maps here are independent. And if they are independent, we have the mechanism for pattern separation in the hippocampus. Then we can keep our memories separate. So how can we explore this question? I have 1 minute to do that. So if we then record cells that are quite small here, so this cell and this cell, and then we have some bigger scale cells, you see the big fat fields here, and then we just do a very simple experiment. So this is when the rat is running in the normal box, 1.5 meter, and then we just push the wall in. And what happens with these tiny grids is that you just chop off the rest of the grid pattern. But in the big grids, you squeeze. So even though it's the same rat, the same brain, these cells react differently to this simple sensoric input, which is to move the wall. And this is just to show it mathematically that you can, if you just stretch it by 5 centimeters, then the highest correlation for these cells are when you don't stretch at all, and you correlate this map with the original map. But you have to stretch it 50 centimeters, which is exactly the size you use to push the wall in for the big modules. So this explains pattern separations. Can I have one more minute? No? Close? Okay. So this is just to say that art, and I had hoped to tell you about emotions and so on, that will enhance memory. But I think I would like to, and I also had something about taste aversion. That's very different types, but we can maybe have questions. And I would also like to invite you then instead to the lab and enjoy the type of research we do. And I hope that I can be invited back to tell more about memory later on. Thank you very much for your attention. No questions?